Hello everyone, welcome to my channel Dr. TAPATI'S presentation. Today topic of the presentation is bioreactors for solid-state fermentation. I'm going to mention about various problems encountered in SSF bioreactor. Then I'm going to discuss about classification of bioreactors for SSF including tray bioreactor, packet bed bioreactor, rotating drums bioreactor and gas solid fluidized beds bioreactors. Then you will get to know about α-amylase production by SSF in different bioreactors. Kindly stay with me till end of this video. You will get to know a lot of new information. A bioreactor refers to any manufactured or engineered device or system in which a chemical process is carried out in a controlled environment by organisms, cell lines, and biochemically active substances derived from organisms. It provides control parameters or environments such as temperature, pH, nutrient supply, oxygen supply to optimize the growth and production of desired biological substances. Various microorganisms are used in bioreactor system during the fermentation processes. Those organisms are either bacteria, fungi, or yeast. Various biomolecules are synthesized in bioreactor system by organisms or cell lines. Popular cell lines cultured in bioreactors are plant cell lines, mammalian cell lines, microbial cell lines, yeast cell lines, and insect cell lines. Bioreactors are used for the production of different types of biomolecules used in medical and pharmaceutical industry, food beverage and other industries, and cosmetic industry. It is used in the medical and pharmaceutical industry for the production of pharmaceutical and medical components like vaccines, blood components, cells, allergens, genes, tissues, and recombinant proteins. In food and beverage industry as well as in other different industries, bioreactors are used mainly for fermentation for the production of different metabolites like beer, wine, cheese, yogurt, other dairy products, antibiotics, aromas, biosurfactants, enzymes, organic acids, microbial biomass production such as single-cell proteins, probiotics, and food colorants, then wastewater and organic waste treatments, biofuels, etc. Whereas in case of cosmetic industry, bioreactor systems are used for the secondary metabolite production from plant parts and tissues. Typically, a bioreactor contains four main components. First of all, a main chamber, then actuators components, sensors, and software to control and monitor the experiment. Main chamber contains the cells or cell-derived metabolites being studied. Actuators components can manipulate and control the environment within the main chamber. Sensors are used to monitor various parameters related to cellular metabolism, such as metabolite levels, pH, temperature, oxygen uptake, etc. Software integrates the actuators and sensors to enable precise control and monitoring of the cellular microenvironment. In a fermentation process, a bioreactor provides the environment for the growth and activity of the microorganisms that carry out the biological reactions. There are different types of bioreactors for fermentation processes, and each design tries to make conditions more favorable for production of different biologicals. There are four major roles of bioreactor. Those are to contain the substrate, to contain the process microorganism, to protect the process microorganism against contamination, and to control environmental conditions to optimize growth and product formation. In this video, as main topic of the discussion is bioreactors for solid-state fermentation process. First of all, you should know what is solid-state fermentation process. Although already I have discussed it in my last two videos, I'm going to summarize the process. Solid-state fermentation is a three-phase heterogeneous process that combines solid, liquid, and gaseous phases to convert a starting substrate to value-added products. It is performed on a non-soluble material that acts both as physical support and source of nutrients in the absence of free-flowing liquid. It has drawn a significant interest in the last few decades for the development of industrial bioprocesses, especially because of its economic and environmental sustainability, whilst producing more value-added products with a decreased risk of contamination. It is commonly utilized in the production of different metabolites, including antibiotics, aromas, biosurfactants, enzymes, organic acids, then biofuels and environmental purposes like bioremediation processes. Bioreactor design is an integral part of biotechnology, and design of an efficient industrial level reactor for solid state fermentation is of significance because it has several advantages over submerged fermentation. Already I have discussed about various advantages of SSF in one of my previous videos. You should watch it. In spite of the potential application of SSF in order to obtain the value-added products, few designs are available in the literature for bioreactors operating in solid-state conditions as compared to submerged fermentation. This is mainly due to several problems encountered under this fermentation condition. Only those microorganisms which can grow at low moisture levels can be used, like fungi. Usually the substrates require pre-treatment. The solid nature of substrate causes problems in the monitoring of the process parameters like pH, moisture content, substrate oxygen, and biomass concentration. Many important basic scientific and engineering aspects are characterized poorly. Information about the design and operation of reactors on a large scale is limited. Scale-up is difficult in this process. The removal of metabolic heat generated during growth may be difficult. In some SSF, aeration can be difficult due to high solid concentration. Spores take longer time for germination, and therefore cultivation time is longer than SMF. Mass transfer is limited to diffusion. The main reason behind designing new SSF bioreactors is to address or resolve major problems associated with solid-state fermentation process, such as oxygen transport, metabolic heat removal, moisture gradients, and the heterogeneity of the solid substrates. Performance of a good bioreactor is controlled by two main factors: the interactions between the microorganism and its local environment, and influence of the design and operating strategies on conditions in the microorganism's local environment. Effective bioreactors have high biological reaction rates, including growth performance, substrate consumption, product formation, and byproduct synthesis rates. Already existing SSA bioreactors can be divided into two general categories: laboratory scale and pilot and industrial scale. Each of the bioreactor systems in its own design tries to make conditions more favorable for production of different biologicals, as well as they have some limitations. Let's first of all discuss about the laboratory scale bioreactors. In this type of bioreactors, from a few grams up to few kilograms of dry solid medium is used in solid state fermentation process, and 1 to 1000 liter liquid medium used in SMA process. These are small scale bioreactors mainly used for research and developmental study in laboratory. They serve as an important intermediate step between benchtop experiments and scale up to pilot scale or industrial level bioreactors. Petri dishes, jars, wide mouth Erlenmeyer flask, rocks bottles, and roller bottles. These are the examples of most popular laboratory scale bioreactors, which are very simple. Due to small-scale operation, these bioreactors allow better control and monitoring of parameters like temperature, pH, dissolved oxygen, etc. as compared to large-scale bioreactors. These are mostly made up of glass, steel, and sometimes polycarbonate and polypropylene. These systems can be highly automated and customized for specific applications and process requirements. These type of bioreactors provide a valuable tool for researchers to study biological processes at a manageable scale before transitioning to larger production environments in industrial level. Once reached optimal lab scale conditions, including inoculum rate, optimum matrix quantities, optimal temperature, pH, etc. Pilot and industrial scale processes are studied and optimized in bioreactors with sophisticated control systems. Based on the presence or absence of agitation and forced aeration, SSF bioreactor design can be classified into 4 groups. First group is unforced aeration without mixing or agitation. Forced aeration without mixing. These two groups are static. Unforced aeration with continuous or intermittent mixing agitation, which is coming under group 3. And group 4 means forced aeration with continuous or intermittent mixing agitation. Example of group 1 type of bioreactor, that is a tray bioreactor. Example of group 2, that is packet bed bioreactor. Examples of group 3, those are rotating drum bioreactor and stirred drum bioreactor. Examples of group 4, those are gas solid fluidized bed bioreactor, stirred bed bioreactor, rocking drum bioreactors. I'm going to discuss about those bioreactors one by one. This figure is showing the classification of SSF bioreactors based on agitation or mixing and aeration strategies used. If you consider about the group 1, in that case it is without forced aeration and without mixing or very frequent mixing. Example is tray bioreactor. Now if you consider about the group 2, it is coming under the with forced aeration but without mixing or very frequent mixing. Example is packet bed bioreactor. If you consider about group 3, it is coming under the without forced aeration and with continuous or frequent intermittent mixing. Examples are rotating drum bioreactor and stirred drum bioreactor. If you consider about group 4, that means it is coming under with forced aeration and with continuous or frequent intermittent mixing. Examples are gas solid fluidized bed, stirred bed and rocking drum bioreactors. Now I am going to discuss about one bioreactor system from each of the 4 groups, including tray bioreactor, packet bed bioreactor, rotating drums bioreactor and gas solid fluidized beds bioreactor. First of all, tray bioreactor. It corresponds to the tray fermenters as typified by the famous cozy process. It consists of a chamber which may be an incubator or a room in which air with controlled temperature and humidity is circulated around a number of trays made of either wood, metal or plastic. Each tray contains a thin layer of substrate, typically between 5 to 15 cm deep, and usually has an open top and perforated bottom. The main disadvantage of this configuration is that this technology requires numerous trays, large areas, and is labor intensive. Packet bed bioreactor is mainly coming under group 2, that means with forced aeration and without mixing. Typically, it involves a static bed on top of a perforated plate through which conditioned or humidified air is blown. The temperature is maintained by external devices like heat exchangers or cooling or heating jackets. The main drawbacks of this configuration are difficulties in obtaining the product, non-uniform growth of microorganisms, poor heat removal, and scale-up problems. Intermittently mixed packet bed bioreactor is also available, in which conditioned or humidified air passes through the bed and an agitation device is periodically used to mix the bed, and at the same time water is sprayed if necessary. The main drawback of this equipment is the need to prepare and inoculate the substrate in other equipment before filling the reactor. Rotating drums bioreactors are coming under group 3, that means unforced aeration with continuous or intermittent mixing or agitation. In this bioreactor, the substrate bed is held within a horizontal or near horizontal drum, which may or may not have baffles, and which is rotated continuously or semi-continuously. Mixing occurs intermittently, continuously or mixed, and without forced aeration. As the drum is semi-filled with substrate, height of the fermenting bed is less, which facilitates good oxygen and carbon dioxide transfer. There are two types of stirred drum bioreactors. The agitation is assured by a rotating drum in the rotating drum bioreactor and by paddles inside the drum unit in stirred drum bioreactor. For a discontinuously rotating drum, the design is identical to that of the continuously rotating drum. The only difference is that bioreactor is rotated intermittently rather than continuously. Therefore, between two agitations, it operates like a tray reactor and heat transfer problem persists. Under these conditions, even with baffles inside the drum, it is very difficult to separate these aggregates. Consequently, the heat, mass, and oxygen transfers are greatly reduced. When the rotation rate of the drum is increased, it can affect the mycelium growth, presumably due to shear effects. There are mainly three types of bioreactors with continuous agitation and forced aeration: gas solid fluidized bioreactors, tire aerated bioreactors, and rocking drums bioreactors. I'm going to discuss about gas solid fluidized bioreactors. A fluidized bed bioreactor is a combination of the two most common configurations, tire tank and packet bed to continuous flow reactors. It is a vertical chamber with perforated plate at the base. Forced aeration is supplied at the bottom chamber at sufficient speed to fluidize the solid substrate particles, which causes mixing of the substrate particles. The mixture of solid substrate particles and gas, which is supplied air, acts like a fluid liquid. An agitator or clump breaker present in this system breaks up agglomerates which can form and settle at the bottom. Due to high flow rates for fluidization, it has excellent heat and mass transfer characteristics. It can be operated in continuous mode, allowing continuous withdrawal of products and introduction of the new reactants. Drawbacks are fluidization requires the use of fine particles and minimum air velocity for fluidization is high in high power requirements. Therefore, large cores and sometimes sticky particles which usually appear in SSF are difficult to fluidize. Although several studies used and set up a wide variety of alternatives to these fermenters, the tray or drum type bioreactor still serves as the starting model to design them, and those are the most popular bioreactor systems used in industrial level. Now I am going to discuss about another bioreactor system, that is spouted bed bioreactor, which is also very popular bioreactor system. It is a type of fluidized bed bioreactor, but in this case, air is only blown upwards through the central axis of the solid bed. In this picture, you can see it is air is blown upwards through the central axis. This is the central axis of the solid bed, which is known as the spout, and therefore only part of the bed is fluidized and the bed can be expanded. Due to the vigorous contact between gas and solids, solids slip down the bottom of the bioreactor due to its sloped sides, and solids cycle continuously. It prevents particles agglomeration caused by high-speed impacts in the spouted bed's core region, which is a big problem in normal fluidized bed bioreactor system. This is more suitable for handling sticky, irregular, textured or sized and large coarse solid particles with different densities and shapes. It has advantages of isothermal operation, high heat transfer rate, reduced gas residence time, which minimizes secondary reactions. The spouted bed bioreactor solves various problems associated with more common SSF bioreactors, including tray and packet bed bioreactors. It shows improved fermentation performance, including higher product titers, yields and productivity, but it has also drawback. Continuous spouting was found to be unfavorable to this SSF, possibly because of shear impact damage to fungal mycelium during spouting. In my last slide, already I have discussed that the spouted bean bioreactor was developed to overcome many of the problems inherent to large-scale solid-state fermentation, including mass and heat transfer limitations in the conventional and tray reactors, and solids handling difficulties seen in packet bean bioreactors. Similarly, Silva and Young observed that Aspergillus oryzae in RRL 697 grown on rice showed equivalent protein and amylase production levels in a packet bed bioreactor system and gas solid spouted bed bioreactor with intermittent spouting of air. All of those activities were higher than those obtained in a tray bioreactor. However, among these three bioreactors, gas solid spouted bed bioreactor was proved to be the best because of without formation of large chunks of rice aggregates knitted together with fungal mycelia. That means without the uniformity and solids handling problems, which were present in case of packet bed bioreactor. However, continual spouting showed negative effect, means decreased protein and enzyme production, and that is possibly because of shear or impact damage of fungal mycelia during spouting. This table is showing different other reports on α-amylase production by SSF in different bioreactors. Bacillus coagulans produced maximum α-amylase activity in aerated bioreactor as compared to large trays. Bacillus species KR8104 produced α-amylase activity of 473.8 U per gram dry fermented substrate in laboratory scale packet bed bioreactor. Aspergillus oryzae IF4300103 produced maximum 19,665 U per gram dry solid in laboratory scale GROWTEK bioreactor. Aspergillus oryzae IF4300103 produced maximum 22,317 U per gram dry solid α-amylase activity in a new bioreactor with two cylindrical glass vessels, one above the other, connected through a standard glass joint and a central connecting tube, which is also a laboratory scale bioreactor. Aspergillus oryzae produced 10,994.74 U per GDS in 600 liter pilot scale fermenter, which is a tray bioreactor. Thank you very much for watching this video. Kindly like, share and subscribe this channel.